start with chapter sexual reproduction in flowering plants, which is very important chapter. So what is sexual reproduction? Sexual reproduction involves two parents, and it is very advantageous than asexual reproduction because it produces variety, right? Variations. So sexual reproduction in flowering plants, and where sexual reproduction takes place? In flower. Okay. So flower, in this chapter we will start flower, a fascinating organ of angiosperm. In this chapter, first we will start with flower, right? So what is a flower? Flower is a modified shoot. Flower is a modified shoot. These are the parts of flower, which is the reproductive structure, sepal, which is green in color. Sepals are green color, which is protective in bud stage. Then petal, which is colored part of the flower, which is colored part. Colored means red, pink, yellow, any color is there. And petal is very attractive to insect also. And insect is needed for pollination. And then this is the female part, stigma, male part, stamen. In this chapter, we will study how this male and female meet. That will study, we will study pre-fertilization, fertilization and post-fertilization. In pre-fertilization event, how pollen grains and ova will form and how they meet, how pollination takes place, and in fertilization, fertilization takes place, and then what will happen to sepal and petal after fertilization. So this we will start from here. So this is the flower, modified shoots develop from floral bud and which develop into a flower, reproductive part of a plant. Androecium is the male part and gynoecium is the female part of a flower. So we will start with male reproductive organ, which is stamen. So male reproductive unit is stamen. It consists two parts. This is anther and stalk-like structure is filament. Okay. Now this anther is bilobed structure. How it is bilobed structure? Just see here. This is bilobed structure. Right? This is anther and this is filament. Okay. So anther filament, and yes, it is one bilobed, and in one lobe two thecca are present. So two lobes are there. So how many thecca? 4, right? So pollen grains, 4 thecca are present, and stamen, male reproductive organ, 2 parts, filament and anther. Filament is attached to the thalamus or petal. Now we will talk about anther, which is dithecous, means one lobe, 2 thecca are present. And what are thecca? Thecca are cup-like structure. Anther is bilobed with each lobe having 2 thecca, and this groove runs separate the thecca. Right? Anther, pollen grains. Next. Now this is one of the most important question which is asked in board examination also. Anther structure, transverse section of anther. So this is the structure of anther. The outermost layer is called epidermis. Inside it, endothecium, middle layers, and last is tapetum. Okay. So epidermis, endothecium and middle layers, these are the protective layers. Why these are protective? Kisko protect karna hai? They have to protect male gamete, obviously. And tapetum gives nutrition to these pollen grain. Okay. So these are the structure of anther. So just see here, epidermis, endothecium, tapetum, and here in between sporogenous tissue are present. Sporogenous tissue are present in between the each microsporangium, right? So sporogenous tissues are present, and these are the tissues which are diploid in nature. What is the ploidy? 2n, right? So here myocyte will be formed, microspore mother cell will form from these sporogenous tissue. Now come to next topic, microsporangium, structure of anther, bilobe dithecus, longitudinal grooves which is separating the theca, right? Tapetum, what tapetum does? It nourishes the developing pollen grains. Now, next topic, microsporogenesis. What is microsporogenesis? Genesis means synthesis or formation of microspore, which is male gametophyte. So, the process of formation of microspore from pollen mother cell through meiosis means will we go here? It will like this, 2N will form N. So this is the sporogenous ploidy is sporogenous tissue, and meiosis will take place, and meiosis what will form? Haploid and microspore. And why we are taking microspore? Because it is a reductional division. So it is reductional division is here. So from 2N sporogenous tissue to microspore mother cell will form. Okay. So come like this, microsporogenesis. Formation of microspore from pollen mother cell through meiosis, which is reductional division. It is called microsporogenesis. Anther develops each sporogenous cell which is inside in microspore mother cell or pollen mother cell. We have seen in anther structure transverse section. So sporogenous cells undergo meiotic division, reduction division to form microspore tetrad, which arranged in a cluster of 4 cells. Yes, it is arranged in a 4 cell, and then at at mature phase it will dehiscize, right? Form as the anther mature, jab anther mature it will mature. As the anther mature, the microspore mother cell dissociate from each other and develop into pollen grains. This is microspore. It will form tetrad, and each will go and separate and form 4 microspore mother cell. Each microspore contains thousands of pollen grains. They are released with the dehiscence of anther. So see the flow chart. How pollen mother microspore mother cell will form pollen grain. Cells of sporogenous tissue, meiosis, reductional division takes place. 4 cells haploid will form, which form as a tetrad, which stay together. And when anther mature, it will dissociate and form haploid microspore mother cell. So this whole process is called microsporogenesis. Now pollen grain, what will the ultimatum, what banna kya hai pollen grain? Pollen grain is the male gametophyte, which is, first we will start with the structure of pollen grain. See, exine. What is exine? 2 layers are there. Pollen grains have 2 layers. Exine, which is external structure, external layer, and intine is the internal layer, right? So pollen grain is spherical in shape. It has 2 layered hard exine and inner one intine, right? What is exine? Exine is made up of sporopollenin, which is very hard and resistant to various like chemicals also, like organic matter, which it's withstand high temperature, acid, alkalis and enzymes. So it can be remain as a fossil also, because it is very it is made up of sporopollenin, which is very hard and resistant to many other enzymes. And yes, germ pores are also present. Here see the germ pores where sporopollenin is absent. Now we will talk about internal layer, which is called intine. It is thin, continuous layer which is made up of cellulose and pectin, right? So it is thin, intine is made up of cellulose and pectin. So see the structure. Achha, this pollen grain is 2 cell stage. Bigger one is vegetative and smaller one is generative cell. So pollen grain cytoplasm is surrounded by 2 cells, which is vegetative and generative. Vegetative cell is bigger. See here, this one is bigger and this one is smaller. Okay, bigger one is vegetative cell and smaller one is generative cell. It has two cell, vegetative and generative cell, pollen grain in cytoplasm of pollen grain. Vegetative bigger and abundant food reserve is present in vegetative cell. And yes, it helps in formation of pollen tube. Okay. So vegetative cell here helps in the formation of pollen tube, and then we will talk about smaller one. This is generative cell, which is spindle shaped with dense cytoplasm and nucleus. One more thing, in 60% species of plants, pollen grains are shed in two-cell stage. So at this two-cell stage, 60% species are shed, where 40% species shed in three-cell stage. How it becomes now three-cell? Generative cell again divide mitosis and it form two male gametes and three-cell stages form, which is shed at in 40% species only. Okay, so see here, divide mitotically into two male gametes. Now we will talk about one more thing, economic importance of pollen grain. Pollen grain is very rich in nutrients. Many countries, they are providing as a pollen tablets, which is used as a food supplement also. Pollen tablets and syrup increases performance of athletic and race horses also. So it gives energy. They are stored for years in liquid nitrogen. They are used as pollen bank and in crop breeding programs also. Pollen grains, yes, some of the pollen grains, they are allergic to some people. You have seen like someone having allergy with flowers, right? In anther या किसी को allergy है, तो pollen grains also have advantage also and disadvantage. It parthenium, carrot grass are the example which are allergic for some people, right? And it leads to chronic respiratory disorder like cold, asthma, bronchitis, etc. Now we will study female reproductive part, which is pistil or also called carpel. Okay, pistil is the female reproductive part of a flower. Okay, yes, we have studied this also. Again, I will just remind you here. This is the female part, which is stigma, style, and basal part. Bulged part is called ovary. Stigma is it is the part which is receptive to anther. Anther will come and land here. So it is the landing part, stigma. And style is the part which connects stigma to ovary. And ovary is the part, bulged part, where ovule means megasporangium or ovule formation, female gametophyte is present in ovary. Okay. So female reproductive have a single pistil, mean monocarpellary, mean single pistil, like in China rose, in hibiscus, we have seen single pistil is there, which is called monocarpellary. And sometime more than one is also present, multicarpellary. If more than one stamen or pistil is present, then it is multicarpellary. Okay, multicarpellary also, they may be two types, syncarpus and apocarpus. In multicarpellary, more than single pistil is present, 2, 3, 4, anything, 10, and the pistil may be fused, that is called syncarpus, and it is free, that is called apocarpus. See here, this is the diagram of NCERT, where these questions also asked that what is this and which type of pistil is this. So this is all pistil you are seeing. There are many pistil which are fused together, and that is called syncarpus. And this is the example of papaver. Okay, and this is the hibiscus pistil. And multicarpellary, apocarpus, where the pistil and the carpels are free. See, all are free, and here it is fused. Fused one is called syncarpus and free one is called apocarpus. Okay, so once again we are going this. Each pistil has three parts: stigma, which is the landing platform where the pollen grains will come and land here, and style is the elongated part, and ovary is the basal bulged part. Okay, I think it is clear. Now we will talk about ovule. Ovary, here ovule is present inside the ovary. Okay, so ovule, now we will talk about ovule, how it is, the structure of ovule. Anatropous ovule structure is there, and in examinations mainly ask, draw the diagram of anatropous ovule, right? In examination, frequently asked question is this, right? So this is the structure of ovule. Okay. So see here, what you are seeing that in ovule, these are the integuments. What are integuments are doing? Integuments are the protective covering, right? And inside where this embryo sac is formed, here nucellus is there. Nucellus is the food reserve part where food are stored. Okay, so these are the nucellus, these are the integuments. Upper one are antibodal cell, middle one are the central cell, and here it is the synergid part. uh, One egg is present and two synergid are present, and here see the integument tip, small opening is there which is called micropyle. Okay, so micropylar end, egg is synergid part is present in micropylar end, and this one is chalazial part where antibodies are there. So we can learn like this: Ca chalazial antipodal, and Sm, S for synergid, and where it is present at the micropylar end. One more thing, micropyle and chalazial are both opposite to each other. Okay, so we have to learn this, like in micropylar end synergid egg part is there, and in chalazial part antipodal is there, and in between secondary nucleus polar nuclei are present. Okay, so mass of cells, the nucellus at the body of ovule contain reserve food material. We have already discussed. Two or more protective covering of nucellus is integuments, which is except at the tip of small opening called micropyle. Okay, so here integument is there, which is the protective covering. Inside the integument nucellus is present where stored food is present, or at the tip where small opening micropyle is. Basal part of ovule is called chalazial part. This is the chalazial part, this is the basal part, and opposite micropyle and chalazial are opposite to each other. Cells of nucellus are rich in reserve food. A single embryo sac or female gametophyte located in the nucellus, which later will form megaspore. So this is the embryo sac which is present inside the nucellus, and this is the structure of megaspore or ovule. Okay, now you have studied in microsporogenesis that synthesis of microspore take place like this in female gametophyte. Synthesis of megaspore, female part will form. So one of the nucellar cells in the micropylar region is differentiated into megaspore mother cell. It contains dense cytoplasm and nucleus undergo meiosis to form four haploid cells called megaspore tetrad. There also you have seen microspore tetrad is there. Like this megaspore tetrad, four haploid cells will form, in which three megaspore degenerate and only one megaspore remain functional. So like that also in microspore we have studied the flow chart. Here also we will study the flow chart. Megaspore mother cell which is diploid to an spelling is wrong. Megaspore haploid monosporic development. What is monosporic development? Means one embryo sac will form from one megaspore, that is called monosporic development. So cells in the nucellus near micropylar region, meiosis takes place, megasporogenesis, four cells are formed near micropyle, one will remain functional and three will degenerate. Functional megaspore enlarged, two nuclei will form, two mitotic division, four nuclei are formed, and then it will like this take place, like this. See here at micropylar end, see the nucellus, one is megaspore mother is present, division will takes place, right? Two will form and then four, and here upper one will degenerate and only one will be remain functional. So this is the formation of megasporogenesis. How it will one functional megaspore. Now what will happen? It is the first cell of female gametophyte. Its nucleus undergo mitosis. Now this is very important, and this part from 2 years I am just observing that this question is coming in board examination from this year also, in 2025, and in previous year also. This is very important. What will happen here? This functional megaspore First cell of female gametophyte, its nucleus undergoes mitosis, and the two nuclei move to opposite poles and form two nucleate embryo sac. Again, two mitotic division takes place, one here, again two here. So how many? Three. In each of these two nuclei form an 8 nucleate embryo sac. How it forms? See, two. Now again this is dividing, one more, 2 and 2 are formed here, and again this divided into 2 and this divided. How many? 8, right? So now after rearranging this part, see, 3 will be here and 3 will be here and 2 will go in central. So this is the synergid egg part, which is present at which end? We have studied na SM, which is present in micropylar end, and antipodal is present at which end? It is present at chalazel end. Okay. So functional megaspore enlarges. Mitotic division, two nuclei will form which move opposite to ends. Now again two mitotic division and 4 nuclei are formed. After this, cell formation takes place. Cell wall will form and egg apparatus will be at micropylar regions. So 1, 2, 3 will go here, 1, 2, 3 will come here, and this 2 will come in central cell, right? So this is the very important topic. This is the mature embryo sac, and this is the frequently asked question ki how many nucleus and how many cells are present in mature embryo sac. So how many cells are there? 1, 2, 3, 4, 5, 6, 7, 8. 7 celled and 8 nucleus are present in mature embryo sac. So this is the microsporogenesis and male megasporogenesis. Now we will start with next topic, pollination. Thank you and have a nice day.